into two sections. First part is the novel trait integration into wheat, how we could achieve that goal since we are having some challenge for food production. And then the second half would be the molecular cytogenetics and plant genome organization. And recently we did a really excellent work comparing the American chestnut and Chinese chestnut satellite chromosome for the ribosomal gene. And you will see that we know when the ribosomal gene in the satellite, we thought it's on the heterochromatic region, but in fact, genes are not in heterochromatic region. That's what we just recently found out. Then we'll talk some today, and if there is an opportunity comes up, we'll talk in details later. Okay, let's go on. Here, let's start with the first two slides, and you see where it stands, molecular cytogenetics today. We all like working with DNA. The main reason is the funding, but cytogenetics is not fun to work. It really takes a lot of effort and patience. It's something like boring science, but there's a great thing one can achieve. What we do, we take the DNA, label it, and put it on the chromosome directly, and to find out where is the marker or is the gene located in the chromosome, whether it's near the centromere or towards the end of the arm. And when you see the exact location of the gene, then you can formulate your breeding strategy to transfer that trait from one line to another line, or one species to another, your cultivated species. That's what it looks like, where it's exactly landing the gene in the chromosome. Here is this example of sorghum chromosome. And interstitial, this is the centromeric position. About 50% of the chromosome is heterochromatic, and you won't find any marker or gene here with this. So when we even do the develop the linkage map, we don't find anything here. And this area, it looks like light blue stain, and it's the euchromatic region. This is on the long arm. The same thing is here. I'll talk later in this seminar. So this slide, not to offend anybody, not any graduate student, that what is the funding situation is taking to us. We hardly, our new generation, know about how the chromosome looks like. I will let all the graduate students read that statement. I won't. So this is not their fault. Our fault is the system. There's no money for chromosome research. This slide is from the human chromosome. So you read that statement. Okay, I'll be talking about more about chromosome. That's why I brought that chromosome issue here. So as you know, that in 3 decades from now, and their population would be about 8 billion. So we need to feed that population, and that's the things comes on the researcher, how we can achieve that goal. And as we know, plant breeding, its risk is peak for a new high-yielding cultivar, but still they are working really hard to get it as good as possible. So now it is time to act. There's no time to wait for tomorrow to make that decision. So how we can achieve with new science? That's new science. I'm talking about GMO and gene editing, and I'm not going to talk anything about those today. So then that also is not my area. I'll be talking about how we can transfer the novel trait through white crossing. That the example would be in wheat. The trait I'm going to talk about, the bread making quality. And then in bread, making quality, that would be two different quality gene, gliadin and glutenin. I'm coming back on that. I will touch base about the invasive pests and diseases and bread, wheat, gene pool, and gene transfer, and how we could transport that novel endosperm protein gene from a wild relative into wheat, and how we develop the substitution line and addition line, and how we could achieve that doing the induced recombination using the 5B system. As you know, that 5B contains the pairing regularity gene that's keep intact all the three genome A, B, and D intact. And then once you transfer the gene, then you need to characterize the recombinant line to see how much alien segment transferred. If we always know it carries the deleterious gene, and we don't want that in our cultivated crop background. Then you do the cytological analysis using the diethyl lines. I'll show the demonstration. And then we need to evaluate the recombinant lines and say some about the conclusion. Here is the invasive pest and diseases, what it could do. You see, an example of American chestnut used to be known as the king of the Appalachian forest. It had a lot of qualities, and no single tree possesses those enormous amount of quality in one species. It grew over 8,000 square kilometers in the northeastern United States, decimated by a dreadful fungal disease. And it's completely wiped out in 50 years and first reported in 1904. And that Chinese and Japanese chestnut brought to the United States just for the beautification here. But you see that it was man-made disaster, but it was not intentional. And some 4 billion trees are lost. And as you know, the Quarantine Act passed based on this disaster to prevent such catastrophe happening again. Here is an example of a Japanese chestnut trees and American chestnut trees. I'll show you in a different slide. Another example of emerald ash borer is an invasive pest, first reported in 2002 and killed, as of now, millions of ashes, threatened some 8 billion ash trees in North America. We work with this breeding program and doing cytological analysis for them, and same for the chestnut as well. Here is in bread wheat. I won't talk details about everything, but just for an example, the stem rust resistant breaking down in wheat, and you see there's a lot of things happening now. There's the fungal is overcoming some resistant gene. And then UZ99 first reported in 1999 in Uganda. That's why the name came as UZ99. It's spreading in the wheat belt full growing area around the world is from there. And it's going to Yemen, then Middle Eastern country heading towards Indian subcontinent. But it's still holding. The breeders is trying to pyramiding the number of resistant gene in wheat background. But look, if it breaks down, what would happen? The yield loss could be 70% or more, or sometimes complete. The top left slide what is showing the effect. So there's there is demand and look for that resistant gene. And I see the gene editing system hold a great promise working here. Dr. Borlaug, as you all know, his dedication and his effort producing that high yielding variety. And he saved the world from famine, and that was that's in 1960s and 70s, and that was a great. The famine was forecasting in Indian subcontinents. At the same time, his high-yielding variety introduced there, and he had a lot of challenge taking that wheat in that region. You all know that. Um, in world wheat, about 80% or more that high-yielding wheat, they contain 1B1 or translocation. This is a fish slide is showing that the R segment translocated in with 1B chromosome, and but this one is not good for quality because second gene. And once you can remove that, that would be probably the great thing using the gene editing system. I think that probably the way to go. I'm not going to spend much time with this slide, but you all know is how wheat evolved through different genome. There's A, B, and D. And B genome from wild is probably lost. Okay, and most of the land race, as we know, is probably gone. And however, the wild relatives hold a promise with these novel traits, could be disease resistant, incest resistant, stress tolerance, and quality gene that could be made available into wheat. And on the left-hand side is and the middle is the wheat, and on the right and left image is showing the wild relatives. This is showing that the accession available in wheat collection that Dr. Skopman did that when he was at CIMMYT. Okay, now this comes, where is that gene pool? If it's a primary gene pool, then it would be easier to transfer the gene. If it's secondary, from the related genome would be okay. But when you go to the tertiary gene pool, then the headache comes, but it can be achieved. Here is an example, the variation of glutenin and gliadin subunits. On the red box on the top is the glutenin subunits. The glutenin, it's about 80% of the total protein gene into wheat, and then glutenin gives the strength and elasticity to the dough, and the gliadin protein provides the extensibility, both important, but gluten is the most. Then what we found out that one U chromosome carries about 80 to 90% of the protein gene of that species. And here, the whole past high molecular weight glutenin subunit that one coded for by in the chromosome 1A and two subunits 1B and 1D. And even though it's a best quality wheat that time, and you see the Umbellata species contains higher subunits. So there is a promise for transferring this gene into wheat. So how you produce the substitution line, and good details, you can use the monotilo line to achieve your substitution, alien substitution line here, and then we go from there. This slide is showing the protein separation. And here you see the from 1 to 6 is the addition line of Agilops umbellata single chromosome addition line. How it looks like, this box is showing that single pair of chromosome 1U in wheat background. This wheat background we used that was the Chinese spring. That's why it's now we're using as a CS. Then 8 1U species, and then line is the Chinese spring endosperm protein. Then 10 is 1U substitution for 1A, this one, and 1U 1B, this one, and 12 is the 1U 1D. You see that 1D subunit missing, and 1B subunit missing. So these are the two substitution line. And this slide is showing them an addition line in which background there is Chinese spring and is a pair of chromosome 42 plus 2 from the alien pair. So now the question, where to transfer that glutenin gene from 1U. This graph is showing on the left-hand side is the yield. And you see, it's drastically reduce the yield potential when it's come to the substitution line, even additional line. But when it comes to the sedimentation volume, one of the determinant of bread making quality, it shows promise for the 1U1A substitution line. So that tells us the gene transfer should be on chromosome 1A. And the other reason is about 40% bread wheat. Dr. Peter Payne started about 200 lines, and 40% of the bread wheat that time showed that chromosome 1A didn't code for this glutenin subunit. So in Chinese string, chromosome 1A doesn't. So let's see if we can transfer this subunit into chromosome 1A, how we can achieve that. This diagram is showing that the gene is close to the centromere. That's from the linkage data. And our target should be chromosome 1A. And how we can achieve it, that's homologous recombination. That is the one I'm going to talk about, but there is a way to do other way: ionizing radiation, tissue culture, cloning, transformation, centrifugation, and homologous recombination. The new science is the gene editing. Here is a recent article I read this week, and this really fascinating one. You can read this one. It's really good about that gene editing story. Homologous recombination, how we can do it, suppressing the activity of PH gene. That Dr. Arnie Sears, he transferred the leprous resistant gene from Agilops elongatum using that suppression as well as nullity line. One can use the deletion line, but it says mutant, but in fact it was deleted from that 5B chromosome. Here is an outline how you can achieve this. This is really a complex one. I will go details. First cross with substitution line with nullifor 5B, tetrafor 5D to remove the 5B system. Then you will end up with 5B, monofor 5B, then try for 5D, then use cross with again with nullity select for the high pairing parent as well as protein separation. You have to select every time that 1U chromosome in your system background. So how this is what happened when you cross this one in this system, this plant, it doesn't have the 5B, and it has 3 copies of 5D or 4 copies of 5D, and monofor 1U, monofor 1A, and there is high chance of get the recombination between these two chromosomes. And when you cross with again nullifor 5D, tetrafor 5B, you want to bring back the 5B system, otherwise that would be, you won't end up with anything. And the target was to get this thing. Well, let me show you again this one. The same thing what I'm using, that's the 5B is coming mono, and 5D would be 2 or 3. Here is 3 copies. But again, cross with the nullifor 5B, 5B and 5D, and then you end up with no 5B system. Here is, you need to bring back the 5B, and then select for the recombinant line, then self-feed, do the disomic substitution line for that chromosome, recombinant chromosome, then cross with the ditylo. Then you started the myocyte. That's whether the long or short arm pairing regularly or not. Then you can deduce that how much segment we transfer in our targeted chromosome. Well, here was the target was 1,000 seed, and then I ended up with 5,600 seed, and we started about 32. For the glutenin and gliadin, we obtained 89 glutenin independent recombinant, and 62 from 1A, 1U, and 27 from 1B. Even though we didn't use that 1B substitution line, however, when you use the 5B system, then 5B system, when it is removed from the system, then it pair with anyone. But most likely would be going with this one. They present at monostatus. And then we evaluated finally with 19 glue line and 13 gliadin line. How we obtained this one? And as I said, if you have aim, focus, you can achieve it. And nothing is impossible, as I believe. What after emasculation, I pollinated 3 times, morning, evening, and the following day, the same spikelets. Then that was the reason to get that success. Then when you have the recombinant line, you do the selfing, you end up with selfing line. So we have some recombinant lines, some are homozygous, some are heterozygous. All the A lines are glutenin recombinant. All the B lines are gliadin recombinant line. Now, we need to do the marker analysis. So these are the markers for 1A, 1B, 1D, 1U. Then after screening those, then we did the TILO analysis with the recombinant line. You do with ditilo, long or short arm, or double ditilo. Well, you can get mass from here. Let me show you another illustration that probably you may get some from here. There's a recombinant line cross with your double ditilo line, and when you see, when you screen this one, you will see 40 chromosome, intact chromosome, to 2 telochromosome for the chromosome 1A, short arm and long arm. If it pairs fairly, then you can say, yeah, the insertion was small. But if that doesn't pair regularly, the long arm is that glutenin gene on the long arm, then it tells us the recombination. This segment is probably the whole arm or is longer insertion. But we can reduce that segment. Here is the cytological analysis. What it shows that the one glutenin line that's with chromosome 1A, then when you use the double ditilo with long arm, it's fairly. When you use the both ditilo for the 1A, look, concentrate with this image, 40 bivalents, and they are ring bivalent. What does it tell us? And if you get in most of the cell ring bivalent, that tells us that it's the background is fixed. And here, what it tells us, the short arm is pairing with the short arm of the recombinant chromosome, and pair also long arm with the recombinant chromosome. What does it tell? It tells us it's probably the smaller insertion. So that's what you achieved, and then you need to look at it cytologically. I know this is hard, and also the cytological analysis, I tell you, it's not the brain. You need the instrument. I'll show you the facility of our lab. If you have the right instrument, you can do it whatever you want, and if you have manpower. On the right-hand side, high pairing, if you see here, there is some quadrivalent. This is one quadrivalent. There's another one. This is another one. That's another one. This is a pentavalent. This pentavalent is with 2 telos of 1B, short arm and long arm. When I was analyzing this slide cytologically, well, I was a student like you people. And then my supervisor said, when you get that slide, call me. He sat down with the same cell whole day from 9 to 5. And following morning, he came to me, said, Nurul, I solved that puzzle. And no, I did not this one. Now I understand what he did, and I can do it. And you see, is not easy. That guy was experienced. And that's what we learned from our mentor. That's the mentor responsibility to teach us. And you see how this is, if you set your mind, you can make it. That's my message. Well, these are the recombinant lines, just show you what you can achieve this with the short arm, with the long arm, with the small insertion. And then this drawing is showing that's the insertion with 1A, 1B, and we had also 2 lines with 1D. That's I already discussed before. Okay, the yield, it's again is not that significant, but with the A line and B line, they're showing some promise. When it comes to the sedimentation volume, and it's really significantly higher than the standard one. So that holds promise. But what we need to do next, we need to do to reduce the segment as possible. So that, yes, we can achieve that. This is the outline how to achieve it. You just cross between 2 recombinant line and end up. But you need to be consistent. If you give up with something, if the challenge comes and it's gone, take the challenge and face it, you can achieve it. There's no such thing that man cannot do. But yeah, some we cannot do, that's in God's hand. That's a different issue. So here we are able to transfer the glutenin and gliadin gene independently into chromosome 1A, and glutenin gene influence the bread making quality. Some blind with gliadin gene with 1U or 1A or 1B will produce more grain yield. Additional work must be needed to reduce that 1U segment to get as small as possible glutenin gene of the alien chromosome. The next phase of my talk is the molecular cytogenetics in plant genomics. Presently, I'm working with southern pine, chestnut, oaks, ashes, elders, sagebrush. It's a lot of work. You cannot do everything, but you can do at least some. And nobody knows everything in science. But I'm still learning. And my wife says that I do not have much in my head. That's why I spend long hours in the lab. So it's I believe her. I don't go against her. That's I learned. So let's move on with this molecular cytogenetics. I use that Pro5S, 435S, tDNA, that transgene, bacterial artificial chromosome, repetitive DNA, retro elements, etc. Here I'm going to talk about, again, it comes about the chromosome, how chromosome looks like. And this slide is showing the lentil chromosome, sporulin chromosome, diploid species, and it's pericentromeric, but is not on the centromere. It cannot be on the centromere. And you see, this is 1993 slide, and I'm learning today from this work. I'll show you some fascinating images what I found or analyzing recently. And this is showing that 2 chromatids, or you can say sister chromosomes of 1 chromosome, and 2 dots. That's what it's showing. There's 1 on each chromatid, and this is the nuclear organizer region, that NY, and that this centromeric position is putative. Okay, let's concentrate with this one. Lentil is 16 chromosome. If we do not have this string of NOR 35 signal, we will end up counting 16. In fact, is one publication claimed that one species of lentil contains 16 chromosomes. That was due to that that they didn't see it. Well, I don't blame them. It's just the preparation. In our lab, The science is not much there. Is most important the how you prepare the chromosome spread on the glass slide. That is the key. Fish, I believe anybody can do it with some teaching, nothing else. Concentrate with this one. I'm not going to come here. It's the same thing. Let's see an enlarged image of this one. You see the two string of this NOR. I never found this in any species. I have been working 30 years in this fish. Look at this. I think you're seeing it, believing it. It's two strands attached with that base of the origination of the 35 base, and the centromeric position is somewhere here beyond that signal, and it's two string, and those two string go with that the NOR that's the next is showing, and this is the two string, and we get that two string signal of 35S RNA. That's really a powerful image. And this can take it to a good journal. Okay, here is an genomic in situ hybridization, that is take labeled DNA from one species and blocking DNA from another species in one background. This is critically, and that right chromosome were labeled, and you can see the yellow showing out all the protein chromosome. And I did mention about the leaf phosphate gene CRs. This is 20 LR24, and this is sitting on the tip of a pair of 3D chromosome. This one is just the background fluorochrome signal. And this slide is showing, you can see each and every chromosome showing the centromeric position. And it's all depend on how you prepare your chromosome spread. That's the key for success. Here is another example of maize trypsicum F1. That's when I was at CIMMYT. That was the main project. Project was to identify the trypsicum chromosome in maize background. And accidentally, I should say accidentally, we found a translocation between maize and trypsicum chromosome. When I first showed this cell, well, if time comes back, then I'll go details on a later seminar. When I showed this slide to a few of my colleagues and showed that, look, this is probably the trypsicum segment introgressed in maize chromosome. Everyone else said that it's probably an artifact rule. Then I didn't say anything to them. I went back to the greenhouse, collected more roots, and did the reciprocal way, opposite way to probe it. Here I use the trypsicum DNA and the maize DNA as a labeled probe. And here I use trypsicum DNA as a labeled probe. And you see this is the trypsicum DNA, this is the maize DNA. And this slide is the F1 of maize trypsicum line. And all this bright blue are the maize chromosome. That was maize DNA was used as a labeled probe DNA. And this one is just showing an enlarged image of trypsicum chromosome. And this is how you see nicely there's two chromatids, and this is the centromeric position and is the diagrammatic representation of that chromosome. Transgene, you can identify, locate where it's located in the chromosome. Now the question is, if it's located close to the centromere, if you want to transfer that segment into your elite line, and as you know, some species are amenable for transformation, some are not. And then it comes the breeding. And I remind you, breeding is not going to go anywhere, it's going to stay. That's the key for new development. It has to be there. And here what happened, it's showing this segregating line is a disomics for that tDNA, but this one is just heterozygous. What would happen? In next generation, it will get disomic for this chromosome, or we can lose this one through the selfing process. This is a tobacco chromosome spread, 48 chromosome, completely separated. There is no overlap, and we do not use the cover slip to spread squashing. It's just you do the digestion, the root tip, and smear it on the glass slide. That's it, and it's pretty simple. Here again, that's I was talking about the heterochromatic DNA in sorghum chromosome 1. If you see that linkage map here, there's 3 markers. We used that map at 80, 81, that's about 82 centimorgan, and 3 probe, 2 go to the short arm and 1 comes to the long arm. And all these markers, they're sitting, and all these fish are sitting in the euchromatic region. When you transfer this, say, segment or one gene, potential gene from a wild relatives of sorghum into sorghum, but if it comes with the heterochromatic DNA, again, we are bringing a big chunk of alien segment. How we can reduce that? Yes, we can, but it takes a lot of effort. But the question is, with our marker, will tell us anything? I think not. So we need to be careful about that. So when you say we will be using marker to identify our recombinant line, yes, markers are great, but at the same time, cytology, if you add the cytology in your program, that will give you the full picture of the work. Here, some work about pine. FR gene, our first target was to locate the FR gene in pine. Here is the fusiform rust in pine. It's a devastating disease. And then when you use that probe, we had an ALP marker. And then that when you use the individual probe or take all this throw, put it on the loblolly pine, it goes everywhere. So then when you put it on the short-lived pine, and it hybridizes on a pair of chromosomes. That's when I was talking with Dr. Nelson, he's the program leader, and he said, Nirul, you know what? Short-lived pine short-lived pine is more resistant than any other pine. So, and it contains more repetitive portion of the resistant gene, and that tells with that cytology. So you can well, it's not going to solve everything, but it can bring some of your unanswered question on the table. This is a retrotransposon. We work with Dr. Casula, and that is its new department, ecosystem conservative biology. That's the new name. I can't remember the full name. That's about, I think this is about 2% of the genome, and how this is an interface of pine, and the blue bright dot, those are the heterochromatic DNA. When you work with Chestnut, and we came to know that Chestnut, we have 10 straight linkage map, and 11th one was distorted. And there is a question that it could be some translocation in the background and send the material to Nirul. If I knew that Chestnut myocyte is that hard, I would haven't touched it. You'd not believe it, how difficult. But crop species, it's 100 times easier than the tree species. But some trees are easy to work with. Pine, not difficult. And poplar is much simpler like crop species. But when it comes to chestnut, you'll be pulling your hair off. You won't have any hair. Client, I'm not talking about you. I'm talking about other people. This image is showing a cross-shaped structure, and this tells us it is reciprocal translocation. And we drew the line over it, and it's confirmed that Chestnut has reciprocal translocation in the background. Won't go details with this. And then we mapped individual Chestnut chromosome with, we assigned individual linkage map with all 12 chromosomes. The interesting feature here is about 25% of the chromosome segment that's involved the satellite and node was not mapped in our linkage map or not reported there because we didn't get the recombination there. So there's no marker to map it. The cytology can help. So we identified that satellite. And next few slides, I'm going to talk about this satellite and RDNA. And the marker distribution is different from crop species. It goes all over the chromosome. This is an example of linkage group B. And then you can see how they distribute. This is the individual image of the bacterial artificial chromosome. And again, I said, if the opportunity comes, we can visit in details this work. Here, that's I said, I'll be talking about the 35S organization and DNA composition in the satellite. This slide, it shows the Chestnut chromosome, 24 chromosome, one major 35S locus and one minor, and one 5S locus. And this is an enlarged image of the major locus. And if you see, the short arm and the satellite is very small, and it's completely covered by the enormous amount of fluorescent green signal. And this what tells us probably this one. I'll show some slide from Mollissima, the Chinese chestnut. That's the original species. The other chestnut species evolved from Chinese chestnut. And you see the accumulation of the copy of the 35S rRNA gene is huge, and it's a short satellited chromosome. And if you see that in Chinese chestnut, this is, we named as CC3, that's, it's called AU copper. And we mark as a satellite SAT1 or SAT2. This satellite SAT2, it doesn't show the satellite section, but it's masked by the 35S gene. You see the organization between these two species, and they pair closely. And then this satellite is showing the heterochromatic region of the satellite. Then we went back to study the prophase chromosome. That's what is the organizational feature of the satellite. Here, this drawing is showing we have also 5S, second 5S locus we identified in Chinese chestnut. And yeah, this one is just the illustration in what happened in prophase or chromatophase, and when it has a heavy mass of segment, and it can easily separate it from the mother chromosome. And this is satellite 2, and this is satellite 1. And these are the two satellites from another line, that is Don Saret. And this two satellite in the system, and then is stressed DNA that's connected with this one, this mother chromosome, and this mother chromosome connected with this one. When I studied that prophase or late prophase or early chromatophase, that you know that condensation, it starts from after the DNA synthesis from prophase to metaphase. And here is, I guess, this mid-prophase chromosome. And this slide is showing that heterochromatic DNA, bright blue with DAPI. And this portion is the light blue. And this is under showing the brackets. And this is the homologs of these two satellites. And this is the summary. Then what it shows, two lines or two exceptions are about the same. And third one has shortest satellite, same as American chestnut. The fourth one satellite, same as the other two accession, and one was a little shorter than the other. And then this one is showing the position of the 35S signal and the second 5S site. And that's what it's showing in the circle. Then we use the F1 line, that F1 between Chinese and American chestnut, 12 chromosomes from the Chinese parent and 12 chromosomes from the American chestnut parent. This slide is showing clearly that the difference between those two major, not two major, that's one major 35S signal between two species chromosomes. Here is clearly showing the satellite, and we use the genomic DNA of Chinese chestnut, and it shows us that this segment is species specific, and it is very bright, and we can see it easily. And you can see that half of the satellite, the 1835S signal, is covering that satellite. The next enlarged image can show clearly, and I drew a line, a red line that's separating the 35S and that and the satellite. The green signal is from the genomic DNA of Chinese chestnut. This is a demonstration of between those two chromosomes, Chinese and American, and it shows the accumulation of the copy of the RNA gene. And this one is showing it's covering the euchromatin region, not the heterochromatin and section. That's what we found recently. And this is just came in accidental, that was not our aim, that what we found after analyzing many cells. And it takes time. And this slide is going to explain clearly that whether the signal or the gene is in the euchromatin of the satellite or in the heterochromatin. That clearly shows that signal. This is probably polar side view, and this is near side view. It's two different from spread. It's showing a little bit heavier signal. That's probably the image captured angle, and that's the position of the chromosome. And also clearly showed a snake eyes, a pair of snake eyes here, and the proximal end of the heterochromatic region or the distal end of the euchromatin. And this one is showing this circle is the heterochromatin and this circle is the euchromatin. Then there is no dispute about where is situating in satellite. This is a complex one. It took me about 2 weeks to come up with all this interface nuclei. And I spent about 4 or 5 days to get a lesson done for this figure. And let me explain only one, just only the middle one, and you will, everyone will agree with me that genes are in the euchromatin region. The satellite 1 and satellite 2 is a partial of the interface nucleus and is encircled with the fluorescent dotted line, and it's on this satellite is attached with the heterochromatic DNA of the satellite, the euchromatin. And this one clearly explains where is the euchromatin DNA of these two satellites. This is the euchromatin DNA and signal of the genes of the 35S in the euchromatic region, not in the heterochromatic region. Okay, as I said before, you need an instrumentation. If you do not have the right instrument, you'll be working days after days and with no result. We have the facility. This is probably one of the best, if not the #1 in the nation in plant science. We have 5 of those workhorse in the lab, and then 2 epifluorescence microscope, one with the teaching head, And what you see, your student can see it from here. That's a great teaching scope. And for the workhorse, we have one, and for evaporescence, one. And we have resources. Anyone is welcome to use this resource. And this is well set up. And I won't mind, even if someone comes in the evening and weekend, I'll be there. And so we have another instrument that's a laser capture dissecting microscope. We haven't done much work, but again, you need people to help. It's not one person job. We achieved some for chromosome spread. We spreading the pine chromosome on a membrane slide, and we can separate it probably most of the pine chromosome from each other. And it's really, we achieved a great protocol for separating the pine chromosome in membrane slide. We are not talking about cover glass. We do not use the cover glass to spread the chromosome on the glass slide. Now I'll take a question, and thank you so much for your patience. If there is no question, then I'll go for fishing and this afternoon. And Hong Ben Jong, he taught me how to fish in Somerville Lake. Thank you, Hong Ben. If you are the.
Thank you very much for a great presentation. Do we have questions for Dr. Faridi?
I have a question. Thank you for a very interesting talk. I have a question about the GLUE1U translocation that seems to improve the baking quality. Do you know the nature of the allelic difference? Is it the protein sequence difference, or it's something else?
I probably didn't get your question, but thing is, it's well, this is not 2021. This work is done in mid-80s, and that time almost no molecular genetic information that time. Well, Michael Gale was doing the RFLP work at that time. As you know, he was one of the pioneers for RFLPs, and I think his team was the first in wheat. Anyway, the subunit, that's what we knew that time. There are two subunits in endosperm protein, the gluten and gliadin. As I said, one gives the extensibility, other gives the strength and elasticity. And those two subunits are really important for bread making quality. And the plant building department used to, that time, developed their lines based on that combination of mainly gluten and subunits for getting the good bread making quality. Is that answer your question? If not, then please.
So Nurul, can I jump in here?
Oh, I mean, no, you cannot ask any question.
Okay. No, I don't want to ask questions, but I just want to make a comment here. You know, this subunit came from the genus Aegilops, okay, which is the same genus of Aegilops toshii, the donor of the pig genome, right? And actually, we are blessed with the pig genome because we enjoy, you know, bread, French bread, and bread without the
Amir, let me interrupt you. We're breaking. I'm not.
Am I breaking for you? You cannot hear me?
Well, I'm not blaming, so you sound like baking.
No. I know you did not use baking. Use SPS limitation. But what I said is that, you know, the bread, you know, most of the bread baking quality came from the pig genome, which is Aegilops toshii. And that genus also contains the same genus of amylulata that you tried to bring into wheat. And so the question, just to build on the question of Sakiku, how those segments differ from the ones that we know, like blue A1 and blue B1 and blue D1.
Blue D1.
Especially blue D1, because blue D1 came from Agilox torchii. We know the 5+10, the 2+12. Now we use a lot of that in the breeding program. So how does this subunit that you described today differ from the units that we got from Agilox torchii, because it is the same genus?
Well, the torchii, as you know, the D genome subunit is the best.
Oh, definitely, yes. Yeah.
Yeah.
And what happened in nature, 10,000 years ago when Durham with torchii.
No,
So that's what nature gave us, randomly. But we can still look into the D genome. Do you think we might have lost some subunits along the way of the evolution of hexaploid wheat? That those subunits
I'm not.
We can go back and look for those.
I mean, I'm not getting your question right, because it's the breaking, the sound. Anyway.
No. I mean, no, forget about the breaking. That'd be good. The question relates to what's in the amino acid sequences. I think Sakiko is running is interested in that amino acid sequences in.
Yeah, I was kind of thinking about that direction, the molecular mechanism of elasticity and whatnot.
Okay.
Yeah.
Well, another thing, let me add one thing, that's the torchii, that's D genome glutenin. One can try to transfer into chromosome 1A, because we already have the 1D and 1B. And to further improvement, I would like to see someone transfer that D genome glutenin gene into 1A, and that could be possible. And for the molecular side, yes, I'll visit with you on a later date.
Okay, thank you.
You're welcome.
I think Dr. Medill had his hand raised. Do you still have a question?
Hey, this is Shu Liu. I just a comment. It's not really a question. So probably for the younger generation, new scientists, they are not very familiar with all those skills like Dr. Faridi presented, beautiful slides and a great presentation. And as he said, he spent so much time in the lab to get those down. Of course, right now, with the new technology, we have a lot more, like with a different kind of platform with molecular markers. We can create genetic map. We can also figure out which part is a allele fragment from which relatives. I mean, with the new techniques, we can do that. But as Dr. Faridi said, we also want to see really on the chromosome, which part, how much allele fragment there is transferred to a cultivar. Like TEM cultivar TEM 12 has 1R, has a right translocation on 1A, replaced 1AS, but we're still not sure really how much. Is it the whole arm or just a fragment of arm? We are not sure, but it was developed by breeders, and then we knew it's a great cultivar, and the 1RS provide the insect resistance like green bug and wheat mite earlier, but then later new biotype came in, and those resistant defeated, but breeders, they are smart. They incorporate, torchii with a new resist gene to the new biotype. So those kind of things. Of course, right now the breeding program or Texas A&M now, they are using more primary synthetics with like more different torsion lines and alien species. So really, it's great opportunity for us to learn and to understand how cytogenetics can help for even right now with the new technology and the molecular markers. So we still need it. Thank you.
Thanks for the comment.
Okay, we have reached the end of our time. So I want to thank you again, Dr. Faridi, for coming and speaking to us, and thank everyone for their attendance. And we will see you in 2 weeks.
Thank you.